Firstly, thank you to Dr. Mohammed for making the trip to be here. Much appreciated. This is a collaboration that has evolved very quickly, I'm pleased to say. Dr. Mohammed, you've had an illustrious career in the regenerative medicine field. And regenerative medicine is a widely used term these days. Please, can you give an explanation of the term regenerative medicine in your context?
Thank you very much, Paul, for having me, and thank you, team M42, for the opportunity to be with everyone here. This is actually an exciting event. So to address your question, the field of regenerative medicine is actually a pretty new field. It has been introduced just like probably 25, 30 years ago, so it's still a developing field. The main goal of the field of regenerative medicine is basically to repair, replace, restore, or regenerate a missing function of human tissues and organs. So the basic concept of regenerative medicine, as it was introduced in the early 90s, focuses on combining human cells and materials together to recreate an organ or a tissue. Now, there are many different approaches, different type of cells, different type of materials that they can be used together with, again, an ultimate goal of recreating human tissues and organs.
Thank you, Dr. Mohammed. As you know, the mission of the Abu Dhabi Biobank is ultimately to power personalized medicine, biomedical research, better treatments, better interventions, better diagnostics. It was evident from our early conversations that we complement one another in our ambitions and capabilities, in that one of the key pillars of the Abu Dhabi Biobank is the stem cell banking, both public and private, which has been running now for approximately 1 year, with a mission of being able to treat in excess of 80 life-threatening diseases with stem cells, some that are very prevalent in the region, thalassemia, sickle cell anemia, and so on and so forth. And I'm pleased to say that's very successful. However, we can go further through our collaboration and make better use of the products that are available, or the materials that are available at the birth of a child. And through your R&D, you've built a successful organization doing just that. Maybe you elaborate on some of the opportunities that present through our collaboration between Humanbiologics and the Abu Dhabi Biobank to develop these products and materials within the Abu Dhabi region.
Absolutely. I think this is really what got us excited to work with Abu Dhabi Biobank, with the idea that there is a gift being donated, which is the birth tissue, and you can actually get a lot of cells out of the birth tissue, trying to understand diseases and trying to better come up with treatments in the future. But what's actually more important, that birth tissue is rich of materials that can be used actually to create human tissues and organs. And to your points about addressing diseases, that birth tissue can provide the cellular component and will be providing the material component. So you can create what we like to call them the mini organs. So we can actually study diseases and develop drugs and try actually to have better, safer, and cost-effective therapies in the future using the same exact tissue that if it's not used for R&D purposes, it would have been actually discarded or thrown away.
Absolutely. And you touch on a key point in terms of R&D within the UAE, capacity building, and fostering those commercial partnerships as well through the use of organoids and alike. Maybe you could expand on the opportunity there.
Absolutely. So the organoids, it's a fancy term really that describes the creation of mini organs. Maybe the science is not there yet to create an organ and implant that immediately into someone, but we are able to create those mini organs. So for example, we can create mini livers, and then when we are actually developing a drug and we want to look at the safety profile or we want to look at the toxicity level of the drug, we can actually test that drug on these organoids. Now, how are we going to create these organoids? So to your points, when we have the donated tissue, like the birth tissue that Abu Dhabi Biobank is working on, we can actually utilize that tissue to extract materials that's going to house the cells, so we can eventually create that mini liver, and we can use it for drug testing. So that's actually a very needed area. A lot of pharma has been moving in that direction. And just actually 4 days ago, the US FDA announced that's actually phasing out from using animals for drug testing. So we are actually heading in the direction that if we gotta develop a drug, we will not test that drug on animals. So what are we gonna test it on before we move to clinical trials? It's literally going to be on these mini organs. And that's actually, again, some of the work that we're going to be working together on and serve a lot of the pharma companies that's actually available here, because they're actually looking for that type of resources that they can actually utilize to recreate mini livers, mini hearts, mini lungs, and the list goes on and on.
That's extremely interesting. If you look ahead 5 to 10 years, what do you see as the trajectory for regenerative medicine and the expanded use of organized at a personal level?
Yeah, I think a lot is going to happen between the next 5 to 10 years. Just 4 months ago, the FDA approved the first lab-made blood vessel. So literally now, there is no need to go to the patient whenever they have stenosis or they have a problem with their blood vessels. There is no problem to go and take, for example, like a blood vessel from their legs and put it like in their chest. Now, literally, you can make a blood vessel in the lab and have it ready off the shelf. When the patient needs it, you just plug it. We are working actually with one of our partners. They are starting their first clinical trial in a couple of months for a 3D bioprinted cornea. So they're going to be printing a cornea and they're going to implant that in patients. So over the next 5 to 10 years, apart from the gene editing, the CRISPR technology, the CAR T cells, we have seen a lot being done. Now, the organoids, it's actually gonna be probably the most hot thing out there in regenerative medicine, because as we stand currently, the success rate of any drugs actually in clinical trials is less than 6% across the board. We get promising results in animals, but the moment we move to clinical trials, we cannot actually exceed more than 6%. And just to add some numbers here, the cost of failed oncology drugs in the US is actually $50 to $60 billion every single year. And we're literally talking about billions. $50 to $60 billion every single year is actually being spent on failed drugs that they are actually shown to be safe in animals. Actually, 96% of the drugs that they are shown to be safe in animals, they fail in phase 1 clinical trial immediately. So there is a lot of time being wasted. There is a lot of money being wasted. So when we have something like the organoids, we actually can screen thousands of different drugs immediately and in a very safe and cost-effective way. And as we had a discussion earlier, the personalized medicine, every cancer is different, every cancer patient is different. So the idea to subject the cancer patient to a broad range of chemotherapies, radiation, when they might not respond to it, but they just go through the suffering, doesn't really make a lot of sense. So the idea that eventually, with the work that we're going to be doing, we're going to provide the resources where pharma companies, they will be able actually to take a biopsy from the cancer patient and combine it with the materials that we're going to be working and developing, and at that time actually test the drug and see which chemo dose work, which chemotype works, if the patient is going to be responding, how soon they're going to be responding without subjecting them actually to the pain and suffering that goes with the chemo treatment.
Thank you, Dr. Mohammed, for a comprehensive answer. What I've learned in my year of being here is that Abu Dhabi, the life sciences sector, is extremely dynamic and has the wonderful support of the regulator, the Department of Health. I just wonder, what opportunities do you see for our collaboration working in such an environment in Abu Dhabi, and what challenges as well? What reservations do you have?
I think there are a lot of great opportunities. That's why I think we are working on this collaboration, because in our relationship, you guys have a lot of expertise around the recovery of the tissue, around the isolation of the stem cells, and you guys have the right resources and the right expertise in place to take the cellular component to the next level. We're gonna come in, we're gonna work on the product sides, and I think as a young market here in the US, there are a lot of opportunities to create actually the foundation for the regenerative medicine and take it with the tissue models, take it with studying the diseases. So not only are we gonna use it for drug discovery, but probably more important, trying to understand some of the diseases that you just described, like the thalassemia, trying to understand really how these diseases, they work, how they actually impact other organs, how they progress, and maybe come up with treatments before they even happen, like, cure and so on. Now, with every young market, there is actually challenges that comes with that market in terms of adoption, in terms of probably having the access sometimes to the talents or the resources. But I think with the relationship that we're going to be developing together, I think we're going to be overcoming this over time as we proceed.
Indeed. And a final question, Dr. Mohammed. In terms of capacity building and education and training, our collaboration would lead to MSCs, PhD scholarships, and development of new scientists in the field of regenerative medicine. I just welcome your thoughts on that.
So again, as I described, the field of regenerative medicine is young. So there is actually a lot of training that is actually needed in that area. But then I think about the field of regenerative medicine, that it's actually a multidisciplinary field, in the sense that there is no degree or there is no training that you will end up being actually a regenerative medicine experts after that. So I think, again, with the training that we will be working on, exchange of talents and resources, I think we will get to, I would say, the ultimate goal of our collaborations and exchange of resources.
Thank you. Yeah, thank you. So many layered opportunities through working together.
Yeah, thank you, Paul.
Sure. So we're almost out of time, but if there are any questions from the audience. I realize this is the last slot of the day, so thank you. You have a question? Sorry.
Yeah, my name is Huda Raphael, and actually I have a question about organoids. How you can test for toxicity, efficacy on a tissue. So I'm just kind of boggled. I mean, it sounds amazing, but I would love to know a little bit more. Thank you.
Yeah, so just to simplify it, let's say we want to try a drug toxicity, right? So usually in the animal studies, we actually look at the liver of the rat or the mouse to see basically what happens to that drug, how it's actually impacting the overall body, because the first actually avenue for any drug. As soon as it enters our body, it actually goes to the liver to basically get detoxified, right? So with the organoids, we're gonna bring in the hepatocytes, the liver human cells. We're gonna put them back in a similar 3D environment, similar to how they have been actually in the liver, so they can actually function normal. And then with those actually micro, and we're literally talking about like, micro liter size actually organoids. That's actually like, the nice thing about them, that you can make millions of them in no time. And then you can actually put that drug there, and you gotta look at what's happening for the hepatocytes. What's going on to the creatinine level, for example, or like, the urea, what's happening to the viability of the cells and so on. So you know basically that this drug is very toxic, this dose is good. And then maybe we can put next to the liver organoids, we can put heart's organoids. And now whenever we test the same drug, we can actually see, yes, it's safe for the liver, but it's actually not good for the heart. And that's actually where whenever we actually take a drug, we always see the contraindication, right? Because like, we know that it might impact here and there. And when you look at it, the next level is to do what we call it organ on a chip, meaning basically that we're gonna have all the different organs in the human body on something that is probably the size of a dime, like literally maybe a centimeter. And that's actually where you're gonna have all the human organs in that location. You put in the drug, and then you get the heart organoids telling you what's happening to the heart, the lung telling you that, hey, I'm not happy with that, the liver is not happy, the pancreas, and so on. So when you do this, you can probably screen thousands of different chemistry for the drug in no time, and at a fraction of a cost for running actually single rat for an animal study.
Thank you. Thank you very much.